Hello and welcome back to the channel. In this part of the video, we are going to discuss about a new topic, that is Operon model. Before starting about this concept, let me tell you that I have many playlists regarding different parts of life science like the molecular biology, evolution, then physiology, and also genetics, okay, and some other parts. If you have any doubt regarding or you want such types of video related to your study, then you can do watch that video, and don't forget to subscribe, like and share among your friends. So let's begin with this new part of the video. In this part, we are going to discuss about the introductory part of the Operon model. Before knowing what is Operon, what is model and everything else, let's discuss where this concept came from. Okay, let's begin. Before that, we need to know what is gene expression. As you all know that DNA contains genes, okay? Then these genes gets transcribed, or the transcription process takes place and is for RNA is formed, okay, or mRNA, to be very precise, mRNA is formed. From this mRNA the translation takes place and polypeptides are formed. These polypeptides can be expressed in different forms. Either it can form structural proteins, or it can form the proteinaceous biomolecules, okay, proteinaceous biomolecules, structural protein, or in the form of int. So by these methods you can see that the gene which was present in DNA is expressed in different form. This is known as gene expression. Okay, what is gene expression? The DNA which is transcribed into mRNA, and mRNA which is formed into a polypeptide, or you can say protein, is expressed in the different forms, either structural protein or proteinaceous biomolecule or enzyme. This is known as the expression of gene. By this you must be clear what is gene expression. Next, moving to the regulation of gene expression. Why I am telling what is gene expression, what is regulation of gene expression? Because this Operon model is a part of regulatory in the case of bacteria. Because in case of bacteria we have two important operon system, that is lag operon and TRP operon. Gene expression regulation is different in case of prokaryotes and eukaryotes, but this operon model is mainly found in case of prokaryotes. Eukaryotes doesn't have this specific operon model. That's why I will be very confined to this prokaryotes regulation, okay, because we are dealing with the operon model, and this operon model is only found in case of prokaryotes. This is to be very clear. So how does this regulation takes place? This regulation can be taking place in the different stages. What is regulation? Means to regulate, means to control, you can say, okay, either switching on or switching off according to the requirement. That is known as regulation. You might have seen that is in the switchboard there is a regulator in the beside the fan switch, there is a regulator. What is the role of that regulator? You can regulate to the 1, 2, 3, 4 and 5 according to your choice. Means if you feel hot, then you can regulate to 5. If you feel cold, then you can regulate to 1, or you can totally switch it off. Okay, and if you want in the middle, you can do it. So what is the role of the regulator? Regulator is according to your requirement, according to be very precise, according to the requirement of the cell or the gene, the function is done. That is known as the regulation. So how does the regulation of the gene takes place? The gene regulation takes place in the different stages. First is the transcriptional level, you can control. Second, you can do it in the RNA time while the processing of RNA is done. Next, during the transportation time of the RNA. Next is during the translation time. So the different stages of the cell expression or gene expression, you can do the regulation time. Okay, now moving to the very important concept of operon. Before going to the main deeper concept of operon, what is operon, what are the structures of operon, we need to understand what is positive control and what is negative control. Why you will be understanding what is positive, what is negative? As I have mentioned you, the what is the regulation? In the switchboard, in the fan, you are either turning on, that is you are switching towards the extreme side, means you are positively, and when you are lowering to the lower end, that means you are negatively controlling, means you are switching off to the lower mode, or you are switching off already. Okay, so what is the role of regulation? Either you can positively regulate or negatively regulate. There is the two mode of regulation. So we are going to see what is positive regulation. Okay, as you see the word positive, what do you mean by positive? You will be seeing that the transcription will be taking place. Thus, the opposite is the negative regulation, in which the transcription does not take place. Okay. In the positive control, the main role is of the activator. What are activator? Activator is a molecule which comes and bind to a SIG segment of the DNA. This is the whole structure. What you are seeing is the whole structure of the DNA. Okay. This is the whole segment. This is the whole segment is of a DNA, which consists of different part. We will be discussing in the next part of the video what is this structural gene, what is operator, what is promoter. We will be discussing about it later, but we need to understand what is regulation. We have already dealt with it, and what is positive and negative regulation. For the positive control, mainly the activator comes into play. This activator plays an important role. Why? What happens in the positive control? This is the operon. Okay, the operon mainly consists of 4 genes. First is the structural gene, first is the operator gene, regulator gene, promoter gene, and structural gene. These are the 4 parts. We will be discussing it in the detail later on. What happens? This activator will come and bind to the activating binding site of the operon, and it will increase the level of transcription. So by this you might be clear that the transcription will be going on in the positive control in a very low level. That is, the transcription is not stopped. The transcription will be continuing, but in a very low level. That is in a basal level. That is in a low level the transcription will be going on. When the activator will come and bind to the activator binding site, that immediately the boost up, the transcription will boost up or increase, and high level of transcription can be seen. In this structure or in this diagram, you can see that this is the activator binding site. Okay, this is the activator binding site. Here the activator comes and bind here, and then what happens? The RNA polymerase will come and bind to the promoter region, and it will go along to the structural genes, and the high level of transcription will be seen. Okay, detail we will be discussing in the next part, how the working takes place. So one of the best example of positive control is Arabinose Opera. This is a type of operon in which it is also found in case of bacterial case. Okay, what you see, that is DABIOC. You must be wondering what are all this. This is nothing but the part of the operon. You can see this DAB. Okay, concentrate to this part. These are nothing but the structural genes. Okay, this BAD, these are nothing but the structural genes which will be getting transcribed into mRNA, and these mRNA will be undergoing translation and will form the following enzymes. That's the role of structural gene. You are seeing this part, that is the I part. I part is known as the initiating site. O is known as the operator gene, and C is known as the gene regulator. Okay, which will regulate this operon. So what happens? This regulator gene, that is the C, concentrate to this C. This regulator gene, that is C, will undergo transcription and will form an mRNA. Undergoes transcription means mRNA will be formed. After translation, the C protein which is formed. Okay, this C protein will not be able to bind to the initiating site. Okay, this does not have the capability of binding to this initiating site, that is the I site. Okay, it cannot bind. Next, what happens? When these arabinose will come and bind to the C protein, it will form a C protein arabinose complex, as a result of which the C protein arabinose complex will come and bind to the I, that is the initiating site. Once this complex binds to this initiating site, then the transcription takes place. mRNA will be formed from each structural gene, different mRNA will be formed, and from each mRNA enzymes will be liberated. From the structural gene, that is D, epimerase will be formed, A, isomerase will be formed, and from B, kinase will be formed. Each enzyme has its different role, which you have been known during the enzymes. Okay, in the enzyme chapter, each enzyme had its role. So by this you come to know how this arabinose acts as a positive regulator, as an activator, which activates the initiating site. Before that, the C protein was not able to activate the operon. But once the arabinose come and bind to the C protein, it activated the performance. Okay, it boosts up the performance, and the structural genes was transcribed into mRNA and mRNA into protein. This is all about the Arabinose Opera. You must be clear with that. Next, moving, what is the negative control? Negative control means, by the word negative, you must be understood that they does not undergo transcription. That is, no transcription will be formed. Why so? Because of this master. Then in this positive control, the activator was the hero. Here the negative control, the repressor plays a very important role. Okay, this repressor binds to the operator gene, as a result of which the RNA polymerase which was bind to the promoter region can't move to the operator region, because we know the basic that once the RNA polymerase binds to the promoter region, this RNA polymerase will move through the operator and then it will transcribe the structural genes. But what happens? This repressor blocks the pathway of the RNA polymerase, as a result of which the RNA polymerase can't move to the structural genes, and therefore no transcription takes place. So by this you come to know how the repressor is negatively controlling the regulation. Okay, how this repressor will negatively regulate. Now you understand one more thing, that is, both positive control and negative control is of again two types, that is inducible and repressible. What is inducible, what is repressible? And one more thing, the example of negative control is lac operon and TRP operon. What is lac operon? Lac operon is a part of inducible operon, and TRP operon is a part of repressible operon. Why so? What is negative, what is inducible, what is repressible, and what is the mechanism of lac operon and TRP operon? To know that, do watch the next part of the video, which will be coming very soon. To clear the concept, please do watch this part of the video. In the next part, we will be discussing in detail first what is operon, what are the parts of operon, and what is lac operon, what is TRP operon, what is inducible, repressible, all that will be discussing in the next part. So this will be a short video, that is for few minutes video in the upcoming series. So do watch it, and if you have any doubt, comment and share as much as you can, because this is a very important concept and also confusing concept. People don't understand this concept very easily. What is when it is switched on, when it is switched off, what happens when it is switched on, what is switched off. I will try to make it very clear to you in the next upcoming video. Thank you and have a nice day.